Hello, everyone. It's honored to introduce LTBR-X as a novel immune checkpoint of tumor-associated macrophages for cancer immunotherapy. These are our research background. This study developed a tumor immune checkpoint discovery platform by integrative analysis. We obtained sequencing data of 1380 patients through TCGA and Jiao databases, from which we screened 51 immune-related genes that were closely verified in multiple databases, and screened key therapeutic targets. Subsequently, we experimentally verified the role of these targets for further validation in patients with lung adenocarcinoma immunotherapy. We first screened out the immune-related genes in LUAD by immune score. The analysis of gene ontology and Qiqi-enriched pathways showed that most of the differentially expressed genes were involved in the immune response of LUAD, and the effectiveness of immune score in screening immune-related DGs was obtained. Then we screened out the credible immune-related prognostic genes in the LUAD cohort and found that LTBR, HSPD1, WHSC1 and TAP2 had potential value as immunotherapy targets. We developed a unique immune checkpoint discovery platform, IMAS, that successfully screened out LTBR as a novel immune checkpoint on tumor-associated macrophages. By immunofluorescence staining, and the evidence found in the TCGA database suggests that LTBR-positive TAMs may be involved in regulating the immune microenvironment. Finally, we further confirmed by immunofluorescence staining that LTBR-positive macrophages were significantly increased in patients resistant to tumor immunotherapy. We found a series of experimental analyses that signaling mediated by LTBR can regulate the expression of chemokines such as CXCL-E and the expression of genes involved in T-cell depletion. We next found, after blocking LTBR experiments in TAMs, that LTBR-positive macrophages inhibited CD8 positive T-cell function by recruiting GMDSC through secretion of chemokines and expression of some T-cell depletion-related molecules. Meanwhile, we found through GSA and Western that knocking down LTBR mainly affected W-Nt beta-catenin signaling and non-classical NF-kappa B signaling. Furthermore, we found that they affected lung cancer progression by regulating the expression of chemokine and T-cell depleted genes by RLB and beta-catenin, respectively. And knockout of RLB can inhibit the regulation of CXCL1 after LTBR activation. Downregulation of beta-catenin attenuates the effects of upregulating PDL-E and others after LTBR activation. Subsequently, we found that β-catenin, a transcription factor of W-Nt signaling, can bind to the promoter region of LTBR, and we also experimentally verified that W-Nt signaling can regulate the expression of LTBR. Further experiments confirmed that LTBR recruits GMDSC and inhibits CD8 positive T-cell proliferation through atypical NF-kappa B signaling and W-Nt beta-catenin signaling. Through the mouse tumor model, we found that knocking out LTBR in macrophages was able to improve the immune microenvironment of the tumor. Meanwhile, our clearance experiments with bisphosphonate liposomes also confirmed the fact that LTBR-positive macrophages play an important role in the tumor progression. We next constructed an immunotherapy strategy for targeted TAMs to deliver LTBR small interfering RNA. And the use of this system can inhibit the growth of the tumor, improve the tumor immune microenvironment, and can block the immunosuppressed M2 phenotype of TAMs. We combined treatment with immune checkpoint inhibitors and found that targeted tumor macrophages for delivery of LTBR small interfering RNA could enhance the efficacy of immunotherapy. Ultimately, results suggest that inhibiting LTBR in TAMs could improve the therapeutic efficacy of ICI by blocking the immunosuppressive activity of TAMs. This picture fully shows the results of our research. These 4 points are the research summary of this paper.